Hey, Bio30s, and today we're going to be discussing mutations as part of our molecular genetics unit. We've been discussing mutations on and off through Bio20, through Bio30, little bits here and there, but just wanted to quickly go over the specific types of mutations, why these mutations occur, and what implications these mutations can have on an organism. So to start off with, what is a mutation? And when we use the word mutation, what is it? Well, a mutation by definition is any permanent change in the genetic material of an organism. Now, a mutation can affect an entire organism, as you'll find out, or a mutation can only affect certain cells in a particular organism, and I'll explain why that happens here in a couple of seconds. Generally speaking, most mutations have no effect on organisms. We get mutations in our cells all the time. I mean, again, all our cells are undergoing or most of our cells are undergoing mitotic division. Every time that mitosis occurs, and we've talked about DNA replication, uh, sometimes mistakes are made. And we talked about how DNA polymerase 3 will copy the daughter strands of DNA when it duplicates, and then DNA polymerase 1 will actually will look to proofread that DNA. But there's usually about 1 out of every billion base pairs, there's going to be a mistake that's made. And I'll discuss the different types of mistakes it can make. But there can be more serious mutations, as we discovered earlier in the course. We talked about things like Down syndrome in the cell division unit, where you can actually get extra or you can remove entire chromosomes from the genome of an organism. So we'll get to all of that today, and that'll be part of our discussion. But just so you're aware, most mutations, when they occur, have no really effect on the organism at all. It's not like you get a mutation and all of a sudden you're going to get, superpowers, or you're going to get a mutation and you're going to die from that mutation. In most cases, the mutations have no effect at all. But I'll explain kind of why that usually occurs, as opposed to sometimes where there are implications where you get a mutation that's going to adversely affect an organism through a disease or something like that. Okay. So there's two types of mutations based on the cells that are affected. You can either have a mutation of a somatic or a body cell, or you can have a germline mutation, which are the diploid cells that lead to gametes. So if there's a somatic cell mutation, that's basically going to occur during the lifetime of an organism. And every time our cells divide in our body, there's always a chance that there could be a copying error that occurs. And if that happens, so let's say I have a cell in my thumb, and it gets a mutation. Maybe it doesn't copy. Maybe it copies an A instead of a T for my nitrogenous base at one part of my DNA strand. So an A gets replaced by a T. That cell now has a mutation, but it's only going to affect that cell and all the daughter cells that eventually it will basically uh, evolve, or I guess divide into. So that mutation might only be localized to a small area of my skin around my thumb. And it's not going to affect every cell of my body, because somatic cells are dividing all over your body, and these somatic cells can have mutations that occur spontaneously. So this is a type of mutation that occurs a lot during the lifetime of an organism. It may not affect the organism heavily, but it's more likely to create mutations during the life of an organism, because it's the stomach cells could develop a mutation, or a heart cell, and then when it divides, all the copies from that mutated cell will also have that mutation as well. If you compare that to a germline mutation, this only occurs once. A germline mutation is going to occur during either spermatogenesis or oogenesis, the formation of sperm or egg, and there's going to be a mutation that occurs in the genetic makeup of one of those gametes, or potentially both of those gametes. When those gametes come together during fertilization, that's where that mutation basically lays itself out. Now, because fertilization leads to a single cell, a zygote, that zygote now has that mutation. So when that zygote begins to develop into an embryo, into a fetus, that means that every single cell in that organism is going to be affected by that mutation. So a germline mutation is only going to occur once in the lifetime of the organism, but it's going to affect every single cell in the body of that organism. And we'll get to a couple of examples here in a few minutes. All right. So those are your different types of cells that can be affected by mutations. Obviously, either body cells or germline cells that lead to gametes. But what actually causes mutations? What actually is something that actually causes a mutation? Or sometimes it's spontaneous, They can certainly occur spontaneously, but there are certain substances or types of energy or organisms even that can actually, if you count a virus as an organism, maybe they're not really an organism. There's the debate still out on that one, but there are certain factors that can actually increase the rate of mutation in an organism. And there are physical mutagens, and that's what a mutagen is. A mutagen is a substance or some kind of an energy source that is going to increase the rate of mutations in an organism, whether it's a somatic or a germline mutation. That's irrelevant. These are just things that are going to increase the rate of mutations wherever they're occurring. There's two types of mutagens, as you can see on the slide here. We've got physical mutagens, which basically physically pound the crap out of the DNA. They break it apart. They damage the DNA physically, and that can actually lead to some major problems depending on how many pieces of DNA are being affected. This would be examples of this would be there are certain viruses that do this. HPV, which people get inoculated for, because HPV, if you get the infection from HPV, it's usually not a major infection, but the virus itself, if it gets into the reproductive system of females, can actually increase the rates of cervical and uterine cancer. So there is a correlation between that virus infecting a female and increased rates of cancer, because the virus, in some way, shape, or form, affects the cells in that part of the female's reproductive system when it invades, and it can actually lead those cells to becoming more likely to become cancerous later in life. So that's why people, or women in particular, are asked to get inoculations for HPV virus, because if you're not infected by the virus, that's one less possibility for contracting cancer. And other forms of physical mutagens are ionizing radiations, UVC radiation, gamma radiation, X-rays, nuclear radiation. Any of the energy wavelengths on the electromagnetic spectrum that are shorter wavelengths than visible light. Again, the shorter the wavelength of the electromagnetic radiation, if you've taken physics or Science 30, you're going to know that the shorter the wavelengths, the more intense the energy is in those wavelengths. And once you get past UVA, which is just beyond the visible light spectrum, you go into UVB, then you start hitting into ionizing radiation at the end of the UV spectrum, which would be UVC, and then going into X-rays, and then of course the most incredibly damaging ionizing radiation would be the gamma rays, which are the shortest electromagnetic waves that we've got. So those are all types of mutagens that will physically damage DNA. There's also mutagens that will actually chemically react with DNA. Now, there's a couple of ways that this can happen. These are, by the way, called chemical mutagens. There's a couple of ones that are a little bit oddball, so I'll explain those ones first. First of all, there are some antibiotics that can do this. There are some antibiotics out there that are listed as chemical mutagens, and then it's one of the risks of taking them, and they're not usually standard antibiotics that you would take, but there are a couple of antibiotics out there that have been linked to being chemical mutagens. But there's also, if your diet is not proper, if you're not getting enough nucleotides in your diet, you can't actually form your DNA properly through DNA replication, because your body doesn't have enough access to maybe nitrogenous bases or the sugars that are required, or the phosphates. If we don't have enough nucleotides floating around, then we can't produce our DNA properly, and that's going to cause chemical changes to the DNA. But generally speaking, chemical mutagens fall into one major category that most people are very familiar with, and that's carcinogens. These are cancer-causing agents, asbestos, cigarette smoke. These are substances, when they enter your body, are going to chemically react with cells to then cause mutations to occur. Now, in both of these cases, the result is going to be some kind of a mutation. Now, I would like to quickly discuss the different types of mutations. Now, there are two types of mutations depending on what part of the genetic material or how the genetic material is being affected. You can have what are called chromosomal mutations, or you can have point mutations. And I'll discuss chromosomal mutations first. Chromosomal mutations are a little more serious, because you're dealing with either deleting or adding extra chromosomes. So in case of a human, we know that our diploid number in a human cell is going to be 2n equals 46. Now, this is something we've talked about already in cell division. We talked about this when we talked about non-disjunction, which was an uneven division of genetic material either during anaphase 1 or anaphase 2 of meiosis. Basically, what we would normally see at anaphase 1 is you'd have your tetrads lined up. At metaphase, you'd have your 4 chromatids that represent your 2 duplicated chromosomes. They're lined up. Here's a sister pair. Here's a sister pair of chromatids. And in anaphase 1, they're supposed to do this. They're supposed to separate. Then in anaphase 2, those 2 sister chromatids are now supposed to separate into 2 individual chromatids. Well, what happens at anaphase 1 if this happens, right? Now, 4 chromatids have gone this way, none have gone this way. These cells that are produced on this side are going to be lacking chromosomes. They're going to be missing a chromosome. These cells on this side, where all the chromatids went to, are going to have an extra chromosome. They're going to be one up. They're going to have it's going to be a number of 2n equals 47. The ones on the other side that are lacking a chromosome are going to be 2n equals 45. We've talked about this. This is what we call monomacies and trisomies. So, for example, we've discussed a lot of conditions, and there are some conditions listed here. You don't need to know any specifically. Down syndrome would be a good one to know. We talked about Down syndrome. That's a trisomy of chromosome 21. So we've got 3 copies of the 21 chromosome. Every other chromosome we have in normal configurations. We have 2 of each other chromosome, which gives us a diploid number in people with Down syndrome of 2n equals 47, as opposed to the normal number, which would be 2n equals 46. A monomacy example would be Turner syndrome. Turner syndrome is a monomacy that affects chromosome 23, which is the sex chromosome, specifically the X chromosome. So this would be affecting women that would have potentially 2 X chromosomes to start off with. Well, if only one of those X chromosomes winds up in the zygote at fertilization, those individuals are only going to have the sex chromosome configuration of X. They're only going to have a single X chromosome, and that means they have a diploid number of 2n equals 45. Now, as you can probably well imagine, chromosomal mutations, they can occur at mitosis, which means that certain cells in your body could potentially have the wrong number of chromosomes. They could affect somatic cells. But generally speaking, when chromosomal mutations occur, it usually occurs in a germline mutation, which is going to affect every single cell of the organism. So like for example, a person with Down syndrome has 47 chromosomes off the get-go. They start off with 47 as a zygote, and then every single cell in their body gets 47. It isn't like someone can get Down syndrome if all of a sudden the analogy I use, all of a sudden I get a somatic cell mutation in my thumb. I'm not going to get infected with Down syndrome. It's not like I can all of a sudden become Down syndrome because that one cell decided to unevenly divide into one cell that had 47 chromosomes and one. You can see the one cell would actually have 45. Even the daughter cells would not be identical to one another. There would be like essentially there would be certain cells in there that would have a 2n equals 45 number for the ones that had the monomacy, and there'd be the other daughter cells and their prodigy, the cells that divide from them, would have 2n equals 47 as numbers. So this, generally speaking, when we talk about chromosomal mutations, that's generally starting from a germline mutation for that to happen in an organism, to at least affect an organism with a chromosomal mutation. Now, most mutations are not going to be chromosomal. Most mutations, and these are the ones that are occurring in somatic cells all the time, and they can definitely occur in a germline mutation as well. These are called point mutations. Now, what I've done here is I've written down a section of DNA. And by the way, just based on our last lesson, we've got 2 triplets here. So technically, if this was a strand of DNA that I was coding off of, right? If I was coding off of this to make a protein, this is going to give me 2 amino acids based on our last conversation about cell synthesis, or protein synthesis. So, for example, that's my DNA. I'm just going to write this in here just because, well, I can. That would then, that code on that sense strand of DNA would then code to mRNA. It would actually transcribe to a piece of mRNA, and that would give me the code UGC on the mRNA. I'm just going to put a line there so everyone can see that. And then I would have GGU. So these codons that are on my mRNA now would determine the amino acids that I'm going to produce. I honestly don't know what they are. If you've got your data booklet in front of you right now, you could easily go into your data booklet right now and find out what UGC, what amino acid that would code for, and what GGU would code for on your data sheet, the one that we discussed as part of protein synthesis. Okay, So this is basically a section of DNA that codes for 2 amino acids that would be in a polypeptide chain. There's all sorts of things that can happen to this strand of DNA, this sense strand that we're coding off of, that could cause problems with the production of proteins. For example, I could get what's called a substitution point mutation, where one nucleotide is getting substituted for another. So in this case, the A is getting substituted for a C. I still have two codons now, but the one codon now is going to be different than it was before. Instead of that codon on the mRNA being UGC, it's now going to be GGC instead. So that would probably lead to a different amino acid being coded for on that part of the polypeptide chain that we're developing. An insertion point mutation basically just means that, as the name implies, I'm inserting an extra nucleotide into there. Now that's going to have ramifications, which I'll get to in a couple of seconds. I could also delete a nucleotide. I could delete a nitrogenous base. In this case, I've deleted the G, so now it's ACCA. I've only got 5 nucleotides in that sequence now, or 5 nitrogenous bases instead of 6. Again, there's going to be ramifications for that, which I'll get to in a second here. Translocation. This one, basically, I'm taking these 3 nucleotides here from the end, and I'm just translocating them, sliding them over to a different location. I'm still dealing with 6 nucleotides, but they've been sort of scrambled a little bit. And there's going to be, again, I'll discuss what that's going to happen here in a couple seconds. Inversion, simply I got my GCC, which is over here. That's actually flipped to CCG. So again, I haven't changed the number of nucleotides. I've just rearranged the configuration of the 6 nucleotides that are in that sequence. And then finally, I've got a duplication point mutation, which basically means that I've got 2 extra nucleotides that have been squeezed in. I have AC that gets repeated one more time. I got ACAC and then GCCA. So again, as you can probably figure out, a point mutation is not affecting an entire chromosome. It's affecting only a portion of a chromosome, maybe just a single gene in the chromosome. It could be something as simple as one nucleotide that gets replaced, but it's not affecting an entire chromosome. It's affecting the genetic code of one specific part of a chromosome. But these are way more common mutations, generally speaking, than chromosomal mutations are. These are happening, again, all the time in somatic cells all over your body as cells divide and make copying errors. These are simply copying errors that are not happening properly in DNA replication. DNA polymerase 1 has simply not proofread the DNA properly, and we've got mistakes that are going on there. Now, what does this all mean? Well, what I've done here is I've starred off a couple of my mutations. I'm going to start off with the green ones first, okay? The green ones, which would be substitution, translocation, and inversion. In each of those cases, I haven't changed my number of nucleotides at all. I started with 6 nucleotides, and I'm ending up with 6 nucleotides. So these sections, these new mutated sections of DNA, can still code for 2 amino acids, and any other triplets that are further down the line are not being affected. Really, we're only affecting either one or both of those 2 amino acids in the polypeptide chain. Nothing else outside of that little section of 6 nucleotides is being affected. So all the other amino acids that are supposed to be in my polypeptide chain are going to be coded for properly. So this means that I'm going to get one of 2 possibilities with my proteins. I'm either going to get what's called a same sense or a silent mutation, which is going to give me a same sense or a same protein. The protein will still function properly the way it's supposed to, because I may not be affecting any of the amino acids at all. Remember, DNA is redundant. If you look at your data sheet on protein synthesis and all your mRNA codons, you know that there are multiple codons that will code for any one particular amino acid. So if I change, let's say, I think there was an example I gave you last time where you had a protein that could be an amino acid that could be coded for by CCG, CCC, CCA. Well, if I replace the last nucleotide in that triplet, it's not going to have any effect at all. It's going to be a same-sense mutation, because even with the mutation, we're still going to code for the same amino acid on that triplet that we were supposed to. Now, in the case of some of these, in all of these cases that I've drawn on the board, these are not going to be same-sense mutations. These are going to be what we call missense mutations, because we've done enough damage on these to actually change the coding for the 2 amino acids. Now, if you want to go in and see what the mRNA codons are for my new mutations for inversion or for translocation or substitution, you can see for yourself that they're going to be different than the amino acids that I originally had told you were going to be coded for off that section of DNA. So what we're dealing with now is a protein that maybe has 2 amino acids that are different, or maybe only 1 amino acid that's different. The protein is most likely still going to function, but it's going to function in a slightly different way. I'm going to give you an example of that here in just a couple of minutes. A topic that we've talked a lot about in biology, and it's going to come up one more time because heaven forbid this disease just keeps turning up like a bad penny in biology, but I'll get to that in about 2 minutes, all right? So all of those ones that are green are considered to be either missense or same-sense mutations. The ones that are labeled blue are more serious. These ones are considered to be frameshift mutations. And what I mean by that is we've either added or we've deleted nucleotides. So that means that the entire strand of DNA that we're coding for to make a protein or a polypeptide chain, the whole line gets shifted over. It's a frame shift. So now, for example, my initial codon here was ACGCCA. Well, because I've added one here, now my first codon is going to be AAC. My second codon is going to be GGC, and you can see the problem. The A now, which was at the end of my triplet on the top, now becomes the beginning of the next triplet down the line. So all my triplets down the line past this point are all going to be messed up because I've added one extra nucleotide, or in this case, I've deleted a nucleotide. Here's my problem here. I'm going from AAG to AAC. That may not be an issue because AAC to ACG might be the same amino acid. The problem now is my next triplet, and I don't know what the rest of the code is, but it's going to start, instead of going CCA, it's going to be CA and then whatever. If this letter was G, for example, over here, now that next codon becomes CAG. That's a problem because all of my triplets down the line are going to be affected. Same with my duplication. That's also going to have an effect that way as well. So you can see that the frame shift mutations, there's always one thing in common about them. The amount of nucleotides has changed. If I change the number of nucleotides, that's going to it's going to lead to a frame shift mutation. And that's going to lead to what we call a nonsense mutation or a nonsense protein. If I screw up enough of the amino acids in a polypeptide chain or a protein, the protein isn't going to work properly. Hence the name nonsense. The protein is probably not going to function at all. So these are much more serious point mutations. If you're going to get a point mutation, you want a substitution or a translocation or an inversion. Those are the mutations that are going to affect your proteins in that section of DNA the least, as opposed to an insertion or a deletion or a duplication, which are going to lead to frame shift mutations. All right, well, I'll give you an example of that. Sickle cell anemia. All right, holy smokes, here comes sickle cell anemia again. We talked about it in Bio 20. I've talked about it here in Bio 30 already. Sickle cell anemia is basically, it is a substitution mutation. Sickle cell anemia, what causes the hemoglobin to misshape, is simply a single nucleotide that is swapped out. In fact, I can point it out to you right here. It's right here on the little red bar there. It is initially in a normal hemoglobin, you would have the code on one of your amino acids being, the DNA code would be CTC, which would code for GAG on the mRNA. That now gets switched from CTC on the DNA to CAC, which basically causes, instead of getting glutamate at that part of the polypeptide chain, that's the normal amino acid, you get valine instead. And that simple substitution of one amino acid, hemoglobin is still going to work, but it's going to be altered. Its shape gets altered, and that's what causes the problems. If you have 2 recessive genes, that's what causes sickle cell anemia. So in some cases, a substitution, a single nucleotide substitution can be all the difference. And in that case, we didn't get a silent mutation or a same-sense mutation. We got a missense mutation, but it's a pretty major one because it changes the shape of hemoglobin. Hemoglobin can still work, but it's going to be radically changed in what it can do and how much oxygen it can actually carry with it, all right? So again, and there's going to be a handout coming out about that here today. That's going to be your homework after this lesson is over. One more mutation that I love this one, and this is a great little story up in northern Italy. There's a little town up in northern Italy called Limone, which is Italian for lemon. They make amazing lemon ice cream up there, and basically. There are 40 villagers in this little town in northern Italy that have a mutation. They have really high levels of bad cholesterol, or LDL, and for whatever reason, they have no issues with heart disease or high blood pressure or atherosclerosis. What's happened in this particular family, they pointed back to some of their ancestors that moved from France to Italy in about the 1600s, early to mid 1600s, and they apparently had a mutation. They had a mutation which allows their HDL, which is their good cholesterol, it's 10 times more effective in their body than it should be normally. So this good cholesterol, even though it's in small amounts, can mop up that bad cholesterol in their body like nobody's business. So this is a very interesting thing for scientists because they want to find out where this mutation has occurred, because this could be something that could be used in gene therapy, which we'll be discussing in the next topic about genetic engineering, where we can actually maybe help people that have more susceptibility, genetically speaking, to heart disease. So this is a mutation that occurred. It's a point mutation. They don't really know exactly what type of point mutation it is, but it is an interesting situation where they've got 40 case studies of this family now living up in northern Italy that have this mutation, and they're looking to see, which chromosome, what section of the chromosome was affected, because there could be real implications there with this point mutation for perhaps maybe solving some of the issues around atherosclerosis, heart disease, heart attack, some very exciting stuff that could be coming out of this family in northern Italy. That's our discussion for today, folks. Takeaway homework for today. I got a worksheet for you, which is called the Sickle Cell Mutation Worksheet. Finish that up. It's going to discuss sickle cell anemia and the mutation, and it'll give you some questions related to mutations in general. That's it for today. Our next topic is going to be the final one for molecular genetics, where I'll be talking you through, here it is, genetic technologies. That'll be our discussion next day, and where genetic engineering is taking us into the 21st century. Thanks a lot for tuning in, guys, and we'll see you soon.